That's just about it. So welcome in, everybody. Thanks for joining. I'm in place of Laka today. Unfortunately, we'll have to attend virtually. Exciting host today. So please welcome in Dr. Robert
Fitak. Don't worry.
He's an assistant professor in the Department of Biology and a core member of the I'm going to struggle with this today. Genomics and Bioinformatics cluster at the University of Central Florida. He earned his doctorate at the University of Arizona studying the genomics of pumas and Mexican wolves, followed by a postdoctoral position at the Institute of Population Genetics in Vienna, Austria, studying camel genomics.
Did you say wolves and camels?
Hey, that's it's I'm reading this and I'm being influenced. Your lab aims to understand the incredible array of biodiversity and specific adaptations in physiology and behavior that makes species unique. So, without further ado, please.
Now it starts turning red after the applause. Well, appreciate the invitation to come here. This is really exciting. It was a very last minute invitation, so you might not have the most recent up-to-date stuff. I kind of said I'll give an existing presentation that I already have. And what's kind of exciting is this is the first in-person seminar I've given since like February 2020, something like that. You know, lots of Zoom ones, but it's kind of fun to be standing up here again. Shall I close the door or?
Okay, but I think everybody is in this room pretty much, so.
All right, and so my lab does a lot of cool and exciting things, or at least to me, but I'm going to focus on one area that we work on that's probably the most relevant, I think, to what some of the audience here would be interested in. That's going to be animal magnetoreception. That's going to be the first half of the talk, and the second half I'll talk about this kind of more exciting stuff, the magneto microbiome is what we call it. I coined the term and I guess I still can't pronounce it, but I like a little bit of alliteration if you can't tell. So I like to start with this graphic as just a giant puzzle. That's because it's one of the things I love to do. So if you went to my house right now on the coffee table, there's a 1,000-piece puzzle. But when you get into the lab with me, I'm really interested in these multi-billion-piece puzzles, right? So it's sequencing DNA, lots of DNA, so genomics, and kind of then the bioinformatics of putting it all together. So sometimes we're using all this data to revisit old questions, maybe to higher resolution, than what's been done in the past. But we're also using this massive amount of data to start asking kind of really new, cool, exciting questions. All right, so just a little bit about what my group does, since we don't just do magnetoreception work, is we do a lot of population and evolutionary genomics, and then we do a lot of things in kind of sensory biology. And so in population and evolutionary genomics, we work a lot with Florida panthers. We use some of these various tools, so new genomes, machine learning resources, epigenomics. And in sensory biology, we use kind of a little bit different set of tools, but particularly in magnetoreception, but we also have some work in neurogenomics of cephalopods, and we do a lot of RNA-seq. And I'll talk a little about what that means. So these are the kind of two flavors of what we work. So magnetoreception. Who here has heard this term before? Some people, 3/4 of the audience. So this is generally what the phenomenon we describe as the ability of animals to perceive magnetic fields, right? And maybe use this information to kind of guide their movements or their navigation. And so, at least before there were smartphones, so maybe the younger people in the audience won't be familiar with this. We used to get around by using a compass, right? You didn't just pull out your, Google Maps or whatever. And we know that it works, or our compass works because Earth has a magnetic field, right? So we can think of the planet as kind of like a giant bar magnet. And yes, with this orientation, the North Pole of the magnet is definitely down near the South Pole of the planet. And so what's cool about this is this magnetic field, it envelops the entire planet. It's been around probably as long as the planet itself. We don't know for sure, but probably at least a few billion years. And it's relatively stable within the lifetime of an organism. So it's really no surprise that many organisms have evolved the ability to perceive this field, right? This magnetoreception ability, and use it to guide their movements. Everything from little tiny bacteria all the way up to a variety of different vertebrates. Our more well-known examples are like sea turtles, different birds that navigate or migrate long distances, as well as fish that navigate or migrate, like salmonids. And there's a variety of other ones. These are just kind of the classic examples. So the big question, though, that still remains in the field is how do they do it, right? What are the mechanisms that underlie a magnetic sense? This is kind of a fun little graphic, but it turns out it, to a first approximation, may somewhat be accurate, or at least some organisms. So basically, we just strap magnets to organisms, and maybe that's how they do it. Just at a little smaller scale. But there's kind of a big problem with understanding magnetoreception, and that is, unlike other sensory systems, so think of vision, think of hearing, we have to have some sort of external apparatus to like gather or attenuate the signal, right? Our eyes, our ears. And basically that apparatus will transmit the signal inside the body into the nervous system. With magnetoreception, it's a little bit different because the geomagnetic field, the magnetic field of Earth, penetrates every cell of the organism equally. And so theoretically, the magnetic sense could be in any one cell of the entire organism. It could be in every cell of the entire organism. And that's been a problem. And we kind of describe that problem as like the search for a needle in a stack of needles. There's the fun joke of the day. And so there are some kind of questions out there that my group works on kind of in this area of magnetoreception, three main areas. First is this, what we call the magnetoreceptor. So what is the physical apparatus? How does it function? So the mechanism, and where is it located? Is it in certain tissues of various organisms, or is it in every single cell? We don't really know yet. And we have this other area we call geomagnetic biogeography. And this idea that animals that navigate using the magnetic field, their distribution around the planet might vary depending on the variation in Earth's magnetic field. So Earth's magnetic field can vary on a day-to-day basis, and every 800,000 years or so, the poles will flip. The North Pole and the South Pole will flip. And we actually study what are the biological implications of that variation in Earth's magnetic field. And kind of lastly, I tried to like collectively come up with a description. I call it consequences of magnetic disruptions. And here's this, if we have kind of unusual events that don't happen very often, so think of things like solar storms and things like human-induced exposure. So an MRI is a really massive magnetic field. Giant cables of electrical current on the seafloor have a really strong magnetic field around them. And how does that alter things like fish behavior? Or how does solar storms affect things like whale cetacean navigation? So these are all various things that we've worked on in the past. So why do we want to study magnetoreception? So I have this little blueprint up here. And when I talk to biological audiences, they usually don't understand, but I have a feeling maybe some people here might recognize this blueprint. I'm hoping. Any ideas? It's not SeaWorld. Good guess, though. Space station, kind of?
I always think station.
Oh, okay.
I apologize. I'm blocking.
Station.
All right, who's a Star Trek fan? Oh, so someone thought of it. This is a blueprint of the USS Enterprise. At least from the fandom. They don't actually, I think, show this blueprint in one of the episodes or so, but the fandom environment has created a blueprint for the USS Enterprise, right? The ship that's traveling throughout space. And what's really interesting here is I highlighted these areas. These are the cetacean navigation labs, right? And so it turns out on the USS Enterprise, there are bottlenose dolphins and humpback whales. You probably can't tell, there's like a bigger whale and a smaller whale. They are commissioned officers on the USS Enterprise, and they are critical for the navigation of deep space. And so there are a couple of episodes, they refer to the whale or cetacean navigation labs. But they're there because we know that they have really great navigational abilities. And by employing them on the ships, it helps them navigate deep space. But of course, the analogy here is by understanding how organisms navigate Earth's planet, we can better understand how we can navigate Earth's planet. And maybe a few years ago, some of you may have seen in the news something like this, right? These videos happening in the UK of these birds crashing into windows. I don't know, can you see over here? Right, pretty crazy stuff, right? I mean, we don't know what's going on with these birds, right? Just coming in these windows, or maybe they're getting lost. Does anybody actually recognize where this came from? I heard it's a movie. I'm not trying to really scare you, right? This didn't really occur. Any ideas?
It's like.
It's definitely an apocalyptic movie,
I think it's the day after tomorrow.
not the day after tomorrow. So it was voted the most scientifically inaccurate movie of all time by a panel of scientists, not Armageddon, the core.
Yeah, Lord, I was gonna ask, I was gonna ask that. You said it's inaccuracy, but.
There you go, that set off a little light and, right? So maybe it's quite scientifically inaccurate, but the premise of this movie, if you haven't seen it, wait, who's seen the core? All right, so if you haven't seen it, the premise is Earth's iron core stops moving. So there's no longer the ability to generate the magnetic field that protects our planet from things like solar radiation, et cetera. But simultaneously, they have a few scenes about, well, maybe the animals that navigate using the magnetic field are getting lost. And now they're slamming into, the buildings and breaking glass, a lot of exaggeration and hyperbole, but kind of exciting, right? So somewhat accurate, maybe. But the most interesting, did any of you notice, I'm gonna go back one to let it go. There's really quickly a fish that slams into a window. And I never noticed it when I actually watched the movie, but it turns out if you take this in slow motion, you can see it. One scene, a fish slams into a window and breaks. And I thought this is exciting because not only are birds navigating what we think using magnetic fields or using the geomagnetic field, but what my lab has done in the past is we use a lot of fish, particularly salmonids, and I kind of thinking that looks somewhat like a salmonid, like a salmon or a trout. And so I thought that was interesting that, yes, they threw a little fish in there just for me because, yes, fish, many different types of fish navigate. I'm sorry?
Not bad.
My point exactly. Maybe it's more accurate than we think. I actually think a lot of the parts of the movie about the magnetic field are relatively accurate. It's what happens when they drill into the center of the earth. Now that's probably gets, I'm sorry?
That's the name, unobtainium. Isn't that the name of the material?
Unobtainium.
That's Avatar.
No, that's Avatar. Oh, well, they have something. They make up some material that, gets into the center of the earth.
Nuclear bomb, right?
All right, well, so I have to move on a little bit. I know it's an exciting movie. You should all go home and watch. I actually bought it so you can borrow it if you ever need it. But the idea here is that what will happen when Earth's magnetic field varies, right? Or maybe it disappears for some reason, and that's not altogether that far-fetched. So we've been measuring the strength of the magnetic field for the last 400 years or so, and there's been quite a precipitous decline. And I don't think the magnetic field is going to disappear anytime soon, but this pattern is reminiscent of what we think happens before the polarity reverses. Before the north and south poles reverse, we know they have a decreasing pattern like this. Now, it doesn't occur regularly, but the estimates are about every million years, and it's been 800,000 years since the last full magnetic field reversal. So essentially, people say we're due for one, right? Is there an impending geomagnetic transition? Now, in order for those poles to flip, by definition, the field has to disappear for at least an instantaneous moment, if not longer. And so we will not be protected from the solar radiation from the sun. And I could go on all day about some of this stuff, but maybe we're not, ready for a reversal to occur. But the whole point is we're interested in studying if this were to occur or to decrease to very, very low levels, what would be the biological implications of that, right? On animal navigation, on sensory systems, and there are other people interested in more the human kind of anthropogenic side of it as well. So if you learn anything doom and gloom about this seminar is that the field might reverse sometime soon, and then who knows, we just might be all out of luck. All right, so again, there's 3 areas that my group works on with magnetoreception, but I'm only going to focus on this area today because I only have about 25 more minutes to talk to you. So mechanism, magnetoreceptor location and function. There's a couple hypotheses on how a magnetic sensor in an animal might work. The first hypothesis is what we call radical pair chemistry or a chemical hypothesis. It's quite complex. Maybe there's some physicists here who can even help me explain it. But essentially the idea is that light is activating or generating a radical pair in certain chemicals or certain proteins, right? A radical pair of electrons. And so those 2 electrons are spinning, and they're either spinning kind of in opposite directions or in the same direction. And the ratio between how often they're spinning in the same direction versus opposite directions can generate two different reaction products. So if they're spinning in the different directions, we get one chemical reaction product. If they're spinning in, did I get that right, the opposite direction, you get a different chemical reaction. And so just the ratio of those two products could tell you something about the ambient magnetic field, which influences those two rays. So really complicated physical mechanism, but there's a lot of evidence now in certain species, notably birds, that this may actually be the case. Another potential mechanism is one we call based on magnetic particles. And these are usually naturally occurring magnetic particles like magnetites, iron oxides. And so this isn't drawn to scale here, but the general idea is that we have a single dipole magnet. So think of a magnet with a north and south pole, and it's attached or associated with a cell like a neuron in some way. We don't exactly know how yet, but this might be like a stretch receptor or a mechanoreceptor. And basically, as Earth's magnetic field changes or you change your direction, this magnetic particle is going to elicit some sort of tension or stress on that nerve cell and induce a signal to the nervous system. Alternatively, these magnetic particles could be like blocking ion channels in kind of nerve cells or in neurons. And as the magnetic field changes or you change your direction, that might move around and open and close those ion channels. And that might transmit or induce a signal, nerve impulse. And so one way that we studied it, and particularly this magnetic particle mechanism, which I'll focus on kind of throughout the rest of this section, is to use a really strong magnetic pulse. And so this is really the best way we can break apart what we think would be a magnetic sensor based on magnetic particles. So I'm showing you this little illustration again of a neuron kind of somewhat attached to a magnetic particle that has a dipole, right, the north and south pole. And if we blast it with a magnetic field that's really strong and really short, like less than a few milliseconds, we can actually reverse the polarity of that magnet. So before the magnet can turn around, like a compass needle, before that compass needle can rotate, we're just reversing it. And we know behaviorally in a variety of species, notably turtles and fish, we know that can disrupt their orientation behavior. Essentially, you can kind of zap them with that magnetic pulse, and you can switch the direction that those individuals want to travel, like when they want to travel on their home, for example. And so, since we know it disrupts their behavior, my particular group is interested in how that might disrupt the genes that are involved in this process. And so again, until I kind of ended up in this field, it was a lot of behavioral experiments, and I started attacking these things with this angle of genomics or new kind of genetic data. And so the first basic experiment is we have fish, control fish, so they get exposed to a mock magnetic pulse. And then we have pulse magnetized fish, right? So they get exposed to that magnetic pulse. And we know from other experiments that I mentioned, it disrupts their orientation behavior. So what happens to the genes that are involved? We specifically selected the brain, and we're going to look at another tissue as well in a minute, the retina. But again, we're looking to see, does this magnetic pulse change expression of genes? Do some genes get turned on and do some genes get turned off? And if so, which ones? And they might help lead us to the mechanism of the magnetoreceptor. So yeah, so which genes are differentially regulated by this pulse? This is done in rainbow trout. The first round in the brain, second round in the retina. And these are two tissues from previous work that we think might be involved in a magnetic sense. We don't know for sure, but there's a lot of work that the neurological system is really important. And particularly, especially if it was a chemical mechanism where light's required, we know that there would be proteins in the visual system that will be necessary. So that's why these two tissues are selected. All right, so a pulse magnetizer, in case you're wondering what it looks like, it's the same kind of machine you would use to magnetize like screwdrivers for your, tools for the VCR. If any of you still remember what that is, you had magnetic parts in your VCR, you would use a machine like this. All it is just a big box of capacitors. And those capacitors just generate a really big electrical current really fast in a coil of wires. So this is just a coil of wires wrapped in like an epoxy, so you can't really see the wires. To make sure this works every day when we pulse magnetize fish, we verify that we can reverse the compass needle. And it's actually a lot of fun to reverse compass needles and then give them to undergrads in a class and tell them to like go find north. And half of them are going this way and half are going the opposite way. And you don't tell them that you actually reverse their compass needles. But it's a fun discussion. All right, so yeah, this is just a basic setup. The fish basically get put right into the center, and the fish or this magnetic coil system is aligned on this north and south axis. So we end up getting a giant pulse either in this direction or in this direction, and we can kind of expose the fish to whichever one we want. Question?
Is the fish in a bowl or is it?
We actually put them in, I don't have a good picture of it, like a little stretcher. And that way the fish stays in a certain straight orientation. We don't want them swimming and turning and rotating. Because we're imagining a hypothetical compass needle in the fish, like in their brain, let's say. And when you put the fish in, that compass needle is going to be aligning north and south. That's just naturally what their compass needle does. And so we want that compass needle aligned north and south. And then when we expose them to the electrical current, we're generating a magnetic field in the opposite direction. So we're reversing that north and south axis of their hypothetical compass needles. So yeah, it's a little stretcher that kind of like wraps around the side of the fish so they stay taut. I don't know if that's the right word to describe a fish staying straight, but keep them straight in the right orientation that we want. I'm sorry? They're in water, yeah. So they are in water inside like a little stretcher.
I did experiments with lactose live fish when I was an undergraduate a couple years ago. And we had them in like tubes, like PVC tubes. So we can like measure the electrodes.
Exactly. That way they can't like turn around, right? Yeah, this would be very similar to that. All right, so what were the results of our experiment? So transcriptomics, so all the various messenger RNA transcripts, the genes that are being turned on or turned off in the brain. So when it comes to the genes, we found a lot of copies of ferritin, and I'll tell you about what that is in a second. And then we had a whole panel of genes that we call just related to visual structures, like vision or phototransduction in some way. But it's interesting because this is in the brain, right? Not the eye. So this was striking to us. So I kind of illustrate both of these here. We had 6 different copies of ferritin, and this is a gene that encodes a protein called ferritin. That's this big thing here. And what it does is it actually stores iron, right? It's just a big cage for iron. It gobbles up iron that's kind of available in the cell, because too much iron is going to cause oxidation and cause a lot of oxidative damage to a cell. So this thing goes around and gobbles up that free iron. And when it gobbles up free iron, it oxidizes it and stores it as various oxidized states like magnetite, a naturally occurring magnetic material. So this is really interesting. It's kind of something we almost expect, right? One of the most important proteins in cells for storing iron. And we know the magnetic poles should be affecting things related to iron. And some of these other genes, we don't have to really go through them one by one, all involved in vision in some way or some degree. And I had to put them in a picture of an eyeball just to kind of remind myself to tell you they're related to vision. But we don't know exactly what they're doing, potentially as it relates to magnetoreception. But one of my undergraduate students at the time, so Monica, kind of revisited our data and looked at kind of a different version of the analysis, look at different genes that are affected. And so here we have our pulsed fish. So every little box here is a fish. And they're split by control on the right and pulsed fish on the left. And darker orange means genes that are turned on, and lighter yellow means genes that are turned down. And we don't have to pay attention to any of them other than this one on the bottom. Sorry, was there a question back here? Okay. The other one I really want to bring your attention to is in the pulse fish, we have a lot of production of this gene called superoxide dismutase, which is actually encodes a protein. And this protein, okay, it'll come back. All right, not sure what happened, but this protein superoxide dismutase, it actually goes around and it's not sharing. See if it's good back up there. Looks good for you.
Yep. If we see the entire screen.
There's always going to be a technical difficulty right in the middle. I don't know, because it looks completely different on the screen. Yeah, make it.
Just doesn't get good.
I know. I'll leave you on a cliff for a minute. Well, it's going to look great for the people on Zoom. And I can't figure out how to move this so it's at least more centered. All right, quick timeout. So if you need water or something, go ahead and grab some.
Close it entirely, restart. Stop sharing. Analyzing their genetic, the speech, sizing, it's like a level of information. So it's just a sort of expression. So the heat maps.
So, like, like not within individuals, but across the individual, like not every.
So that's.
Is that number of person measuring it or across the whole? So each.
Like, I mean, it's like.
Getting half delivered.
That'd be.
Then it would be 100% would be red, 70% would be dark, but now it's not, yeah. Interesting.
It's fun.
Like what we do.
Yeah. Okay. I don't know how to make a comment, but we do it. Apparently the people at the Arboretum were saying that like blood is like the.
No,
They go out and like collect, you know.
Yeah,
bugs
they
and
play
things.
bug bingo.
Oh, love that.
To do. That was my last lecture.
Yeah.
They pick an order, they sign up for an order, and then they all pick it up. But sometimes three people can- they have to coordinate.
Is it what you said Wednesdays?
The first week on Monday.
Changed it.
We start with like half days. Yeah,
That's smart.
Somebody's
and the, you know,
Oh,
a lot of this, you know, we're having, yeah, you've got to describe the diversity. Yeah. To like you do for the but yes, and the way that they do it, that's not too unfair. You know, because
Six.
some, and some people can get bad.
Probably take on it. So.
Yeah. Actually, what I do is I make it. So,
I don't know, you have a fingernails.
class order, you know, then they have your characteristics, and then. So. Major.
That's smart though, they can figure it out.
So you give me some like major characteristics, lungs,
Wait for the host to let me back in the Zoom meeting.
and so lungs tells you that it's like a lung tissue.
Yeah.
You know, that, you know, there's some aspect about it. Special. Unique or anything. Yeah, that's smart.
Stuff.
Yeah.
Should I just put the Zoom or just for them?
My, physiology class.
I'm exposing them.
Okay.
Changes the other day. There's like mosquitoes, or like, they're just switching their vision.
But,
Like, on it. And that would take sense, like. stuff.
Yeah.
So.
Oh, so she go around with you guys. Oh, yeah.
Testing, are we back? So, real quick, superoxide dismutase, it gobbles up free radicals of oxygen. And so the whole, I'm just going real quick here, the whole point of this was we have iron storing proteins being affected, and those proteins are gobbling up iron to prevent oxidative damage. And so if we're screwing up with those or maybe releasing the iron, then we're going to generate oxygen radicals that need to be cleaned up. And that's what this protein does. And so that's what we're kind of thinking right now is we're not actually sure if we've affected a magnetoreceptor or if we've actually just generated oxidative damage by essentially maybe ripping iron out of its storage cage inside its cells. So part of it related to the visual structures, right? We have these genes involved in vision, but we're looking at the brain. So what does that mean? So we decided to repeat the same experiment, but only focus on retinal tissue. Right, so what genes are turned on and off in the retina? And so we have googly-eyed fish. And we first want to know, is there a difference just generally between the left and right eye? We call that lateralization. So do they express genes differently? Because we need to know that before we make our comparison. And then we have our pulse magnetized fish. We also want to, and then the first thing we do is compare just the left eyes between the fish. And then we're going to compare just the right eyes between the control and pulse magnetized fish. And I want to thank the person who really helped with dissecting retinas, Lori Schweikert at UNC. She's now at UNC Wilmington. She's an expert at removing the retina from an eye from a fish, which is great because I would have no idea how to do that. And from our paper where we published this work, we acknowledge a really important thing involved. So Rudy provided the retina dissecting equipment, and I always just make sure I thank Rudy for his participation in this study. It turns out a cat whisker is the ideal kind of tool to scrape a retina out of an eye cup. And the right amount of flexibility, the right amount of strength, that there's nothing you can buy to do it. So we had a friend provide some whiskers, and we use those to remove the retina from the eye cups of the fish.
For your second.
Yeah, well, I mean, they were not plucked from the cat. I can tell you they were shed naturally, and we collected them and used them. So Rudy's been acknowledged in our publication. All right, so to kind of just summarize here real quick, all these comparisons, right? Left versus right eye, left of control versus pulse magnetized fish, etc. We really don't find any genes affected by the magnetic pulse, right? There's several comparisons here and nothing seems to be changing. There is one comparison we made, we get one gene that's a lens protein, but in the end, all these comparisons just turn up nothing. And so really what this told us is that the magnetic pulse is affecting stuff in the brain, but not the retina. So maybe we're starting to narrow down the location of a magnetoreceptor. And the other thing as it comes to visions, maybe we've affected something related to vision in the brain. And fish and a lot of organisms actually have photoreceptors in the brain. They're called deep encephalic photoreceptors. There's essentially an eye in the brain. It's sometimes called the parietal eye or the pineal gland. And in fish, it can basically stretch up to the top of the head, and there's a light-sensitive structure right there, and it feeds down deep inside the brain. And so we're curious if maybe we're affecting something related to this pineal gland or this parietal eye. It is known to be involved in circadian rhythms, so sensing basically the presence or absence of light. And so the next steps that we don't have anything to show you yet is working with Dr. Schweikert. She knows how to dissect this piece of particular tissue out and try to figure out if anything interesting is going on just in that one tissue. And the last thing kind of summarize here is this question of oxidative damage. Again, we still aren't sure if we've damaged a magnetoreceptor or literally just damaged entire cells with this oxidative damage. And we're still trying to clarify the difference in those effects. But in order to see if this is something specific to fish, or maybe it's more general, we decided to look at other species and something actually quite different. And so we looked at spiny lobsters. Has anyone ever been like spiny lobster hunting, or do you call it fishing? I don't know if you call it fishing. But yeah, so Florida is really popular. Certain times of the year you can harvest spiny lobsters. Quite tasty if you like them. But what's great is they have a well-described magnetic sense from a lot of different behavior experiments. And it turns out they do really cool migrations by basically forming a conga line like this and march across the seafloor of the Caribbean. And it turns out they pick their directions very carefully based on the geomagnetic field. So we essentially did the exact same experiments. We looked at 3 different neural tissues. There's the brain, which is somewhere around here. We have a thoracic ganglia, so essentially another cluster of neurons in the middle, and then a subesophageal ganglia kind of in between. And those 3 ganglia are the ones that we looked at, these 3 kind of neurological structures. And from this Venn diagram, there's a lot of genes that are affected, some specific to each tissue, but we were really interested in which genes are affected in all 3 of the tissues. And there's over 300 or almost 400 different genes affected. And rather than naming all of them, we have several that match up with what we see in the trout. Basically, we have several things involved in storing and transporting iron or other metals, and some other ones involved like superoxide dismutase again, which is in cleaning up oxidative damage. So the take-home message from this, if we compare it to a little kind of diagram of the rainbow trout, is we get similar type of genes affected in a similar way, which is kind of interesting. So maybe we're onto some general effect, whether it's again a magnetic sense or not. We see effect in the neurological structures. So again, the brain or similar type of clusters of neurons in the spiny lobster. And potential oxidative damage. So the last problem we still can't fix is separating out these two effects, right? Effect on a magnetoreceptor versus the effect on generally oxidative damage. So any questions on that so far? I'm going to switch directions a little bit here for the second half of this talk. Maybe I'll go a little faster than half, but
It's very interesting.
well, it gets crazier. Hopefully some of you are curious, what does this mean? The metazoan magneto microbiome. So metazoan, anyone know what metazoans are?
Very old.
Very old, but we're metazoans, it's basically animals, right? So pretty much multicellular animals. Magneto microbiome. I'll tell you more about that in a second. So there's a new hypothesis that some colleagues and I have kind of been working on how a magnetic sense might exist in an organism. And it's still technically based on magnetic particles, but this idea that there may be magnetotactic bacteria that are living in symbiosis with a host. So basically, an organism like a sea turtle might get their magnetic sense from bacteria that make magnets. So has anyone here heard of magnetotactic bacteria? The people I talk to. So they're literally bacteria that just make magnets like you would find on your refrigerator, right? Just a little bit smaller. But yeah, magnetotactic bacteria, there's a variety of different types, and they do some really, really cool stuff. So magnetotactic bacteria make little particles of magnetite, right? Magnets basically, little crystals of it. They're just usually a few microns in size. And they store it in a membrane that we call a magnetosome. So if any of you ever learned that prokaryotes of bacteria don't have membrane-bound organelles, these are one of the exceptions. An organelle devoted to storing magnets. So these bacteria can make basically magnetic crystals of a variety of shapes. Naturally, these magnetic crystals stick together and form a chain because the north and south poles of the magnets come together, just like you buy magnets at the store and stack them together. And these bacteria actually take in iron from the environment and kind of oxidize it into these magnetic crystals inside this membrane-bound organelle that I mentioned that we call the magnetosome. So it's pretty cool. There's a whole set of genes devoted just to that process. And these bacteria we actually find everywhere. They're in all sorts of kind of aquatic environments, all sorts of sediment, soil, water. They're ubiquitous throughout the environment. And there's a lot of researchers studying these bacteria for a variety of reasons, because mostly we can control them, right? Using magnetics, we can control bacteria like this in medicine, in industrial applications. So there's a lot of cool work going on with these bacteria. What we're interested in is we were kind of thinking, well, maybe these bacteria, which have their little chains of magnets, which they actually use to navigate, right? I showed you in the very beginning a picture of a bacterium with that little kind of black chain of magnets. These bacteria act like a kind of mechanical compass needle. They're forced into alignment with Earth's magnetic field, and when they swim, they can only swim in one of two directions, depending on their polarity. And it helps them basically find better mud. And so we were thinking, well, maybe these bacteria are actually communicating with a host, so living, for example, on a sea turtle or in a sea turtle, and communicating magnetic information to their host. And so we call it the symbiotic, I don't even remember the symbiotic magnetoreception hypothesis. And it sounds pretty out there, but then a few years ago, someone actually was studying these clams, a northern hatchet shell, and was taking images and reported these chains of magnetite crystals in some of the cells and was wondering, what are these doing here? Maybe it's just contamination. You know, magnetite crystals are kind of also found ubiquitously in the environment. And more recently, kind of after we published our initial hypothesis, a group studying a protist called a euglenozoa, so a single-celled eukaryotic organism. It turns out it's enveloped in a biofilm of magnetotactic bacteria. So all these little strands you see here, these little strings of spaghetti, all of those are individual magnetotactic bacterial cells, each with their own little chains. I don't know, you probably can't see it. Here's one of them zoomed in. It looks better on my computer. That's a little chain of magnets that bacteria makes. So biofilm of magnetic bacteria. And that gives this organism, this protist, a magnetic sense. So they can now swim in directions relative to magnetic north. So pretty exciting stuff. So maybe we're onto something. Maybe these bacteria could be involved with other types of hosts besides just a protist. And so we're thinking of describing a new term, and that's this term we call the magneto microbiome. And we refer to it as the collection of microorganisms in a sample that can biogenerate their own magnetic particles, right? So usually it's magnetite. There's a couple other particles that can be magnetic, but it's pretty much all magnetite. So yeah, microorganisms that make magnets living inside other animals, for example, turtles or fish. So look at this question. The first thing we did was a meta-analysis of 50,000 microbiome data samples. So microbiomes are just studies where they look at all the different types of bacteria in the gut or in the fecal samples or on the skin. They just describe the diversity of bacteria in those samples. So we summarized 50,000 studies or 50,000 data sets. And we looked at how many times we actually found evidence of magnetotactic bacteria, which were generally ignored in all those studies because they didn't care about that. They care about ones involved in digesting cellulose and things like that. So we actually found in microbiomes that are associated with the host, about 40% of them had MTB, which is short for magnetotactic bacteria. It's kind of a mouthful to say. We found it really common in water and sediment, right? That's where we know that these are found regularly. In the soil, same thing, pretty common, so relatively large blue bar. But I also like to point out that in about 40% of human samples, we find magnetotactic bacteria. So if you learn anything from this presentation, go home and realize that there's a good chance that you have bacteria making magnets inside of you. Sorry, what's that?
I'm learning a lot of things today.
And so one thing we're interested in this kind of huge meta-analysis is, do we find certain types of magnetotactic bacteria in certain types of hosts? Is there a host-specific signature? So all these rows here, you can't read them, they're just different species of magnetotactic bacteria. And if there's colors over here, it means that that bacteria occurs in those samples. And the darker the red, the more often it is. And the message here is that you see that there's only like one species of bacteria that's really common in bats and whales, which are both mammals, right? There's just a nice kind of red line going across. But if you look at something like sea turtles or reptile, we have a very different magnetomicrobiome community, right? There's several different species present, usually the same ones in every different sea turtle sample. And there's some unusual ones like this one here that's in red. But generally here is, yes, maybe there is a host-specific signature. So mammals seem to be a little bit different than sea turtles. And this one is just for fun. If you look at all the human samples, there's tens of thousands of human samples. There's like 4 or 5 species that we actually find of magnetotactic bacteria in humans. And again, they're found in about 40% of the human samples. Question in the back.
Yeah, did they ever produce an abundance high enough to measure both in the body, or is this all?
An abundance of the bacteria or of the magnetic particles? So yes, we can measure them using genetics. We can essentially detect the barcodes of these bacteria, which is what we're summarizing in the study in the gut samples. No one has ever yet isolated living magnetic bacteria from any animal sample, which is one of our goals. There's another study kind of based on our work that looked at an association between the magnetotactic bacteria in a gut microbiome from humans and the amount of magnetite in the brain. And there are links between magnetite in the brain and various cognitive disorders like Parkinson's and Alzheimer's. And so there is some interesting work potentially happening there, but that's very, very at the beginning right now.
A sign of a disorder or less magnetite in this kind of disorder?
I'm not an expert. I believe it's more magnetite is, so positive correlation, more magnetite, increased risk of cognitive disorders. And those same people where they studied had higher abundance of some of the magnetotactic bacteria in their gut samples. I mean, we don't know if the bacteria are traveling across the blood-brain barrier, but maybe there's something happening with the magnetite, at least. Again, we don't know. There's only been one study on that so far, but it is interesting and it's giving us some good angles we may want to pursue. So the last part of this is we wanted to scale it up, right? We looked at about 50,000 microbiome data sets. We're trying to think of how could we add to this to get a better picture of where to start looking for these magnetotactic bacteria when it comes to what host they might reside in. So this database is a public database called the SRA. It's the largest genetic database in the world, right? Almost every single study that produces genomic data or genetic data deposits their DNA sequences in this massive database. And I'll kind of describe to you how big it is in a second. And so it's so large that you can't just easily access it, but there are some new computational tools that were recently released that we took advantage of to kind of summarize all the data in that database. But the idea behind this is if you take any sample, whether it's from a sea turtle or maybe it's a blood sample from one of you, and you isolate the DNA from that sample, it will be almost all human DNA. But there's a few pieces of DNA in there that will belong to the microbes and the other organisms that might live in that sample, including contamination. Right? We take a tissue sample, most of it are green turtle cells, but we have occasional bacteria or virus or so that will end up in that sample of DNA. And so when we do all the DNA sequencing, occasionally we capture a sequence that actually wasn't from the host itself. So actually from the sea turtle in this example. So essentially every single genetic data set ever produced is a microbiome sample or a metagenomic sample, which means we're getting multiple species, not just the target. And with all that data deposited in this huge database, we wanted to access that. You can do it visually. We could right now go online, pick one of these data sets, for example, one from a set of DNA sequences from a retina of a blue spotted stingray. And every single DNA fragment in that data set in this huge database gets assigned a taxonomic origin. So if that sequence is unique to a specific organism, we can assign it. And you can see this summary here, you can't read it here, but it summarizes the distribution. Like 90% of these sequences, maybe 95% of the sequences in this data set come from the blue spotted stingray. And that's all this red here. And there's a small component of these sequences that come from the bacteria, again, and the viruses, et cetera. So we essentially mine this entire database for these little pieces of the pie in every single data set and figure out which ones matched magnetotactic bacteria. So we're kind of indirectly detecting these MTV. So we perform this on 26.1 million data sets and over 80 quadrillion sequences or DNA bases. So quite a huge database that we searched. 359 million fragments of DNA, or sorry, 359 trillion fragments of DNA. So we're trying to see how many of these 359 trillion DNA pieces were assigned to magnetotactic bacteria. And this was help that kind of started with an undergraduate student. So Liz Bogg, she's now at University of Washington, doing great. But we use a new search tool, a database searching tool called Google BigQuery, where we can programmatically access those 80 quadrillion DNA base pairs. This is what it looks like when you access it. It's actually really simple. There's just give me every single data set that matches one of these known species. So it's really just maybe 30 lines of code, and we can access that entire database. So the take-home message here is 26.1 million projects. We found 2.7% had evidence of a magnetotactic bacteria in them. Some of those are from environmental samples. Some were from animals. So about 140,000, this is actually an outdated number, it's more than that now, came from metagenomes, which are the samples from the environment. So we expect to find MTV there. But we found at least 60,000 animal samples that had magnetotactic bacteria in it. And there are two magnetotactic bacteria that are relatively common, these two different genera here. And over 396 or about 400 million pieces of DNA that we could assign to magnetotactic bacteria. So a lot. And we're actually still trying to figure out what to do with this entire data set that we've collected. Some things are what we expect. So if we look at species we know have magnetic sense, like spiny lobsters and sea turtles, we find certain magnetotactic bacteria that are very common. One called magnetovibrio in sea turtles, another magnetovibrio and magnetospirillum in spiny lobsters. Does anybody know what this is? Pill bug? Not a pill bug. It's a mollusk, or sorry, it's a gastropod mollusk. But you're on the right track. Yeah, it's called a scaly foot snail. they live at the bottom of the sea near some of the banks and stuff you're talking about. They have an entire armor of magnetite. So all this black here is they deposit magnetite to armor themselves, basically. I don't know exactly why. But what's interesting is it's made of iron, it's naturally magnetic, and we find that they have a couple different types of magnetotactic bacteria when we sequence DNA from them, right? So there's some of these MTB in association with these mollusks or these gastropods that make an armor of magnets. We tried to summarize this by different types of organisms, and really what it is here is we find more, kind of proportionally, more of these MTB in reptiles than anything else. And sea turtles are one of our best models of a magnetic sense. So maybe there's something specific about sea turtles, and birds didn't come up as common, but both have really good navigational abilities. But we think sea turtles and other reptiles might use magnetic particles, versus birds we think might be more often using a chemical mechanism that I mentioned at the beginning. So this is all we have right now. We have an initial paper describing the database, but now we're also kind of trying to figure out what to do, what type of hypotheses and questions we can use this huge database to answer. So going back to that blue spotted stingray I showed you a second ago, there was a sample from the retina, and we actually can detect magnetotactic bacteria in there. But why are they there? We're still working on that. This is the new exciting front of this magneto microbiome that we're describing. So the ongoing work right now, I have one grad student, Juliana. She's actually taking swabs of sea turtle eyeballs. Well, basically lacrimal glands, tears. We're collecting sea turtle tears. Don't worry, we don't make them cry or anything like that. We just literally just rinse their eyes when people are collecting other samples. Like, I mean, people are going in and collecting like fecal samples and like cloacal swabs. This is easy. We just rinse their eye with some gentle salt water, clean salt water, and then we swab them. And we try to isolate magnetically these bacteria and then sequence their genomes. And it's ongoing right now. Juliana's been collecting. We have about, what do we have? Well, we're only allowed 12 nesting females per year. So right now we have 12 nesting female eye swabs, and we have about 50 eye swabs from offshore juvenile sea turtles. And so we're starting to put together a data set. Preliminary data, we do detect magnetotactic bacteria in some of their eye samples. We're just still trying to figure out what the patterns are. But maybe next year you'll hear more about that. But pretty exciting. Some potential work I've proposed, this is kind of, if we get funded, this is one thing we want to do, is we want to work in spiny lobsters in kind of an experimental setting. And essentially, if we treat them with antibiotics that might target types of magnetotactic bacteria, can we disrupt their behavior, their navigational behavior, right? And we can also disrupt it not just with antibiotics. We can use a tool called morpholinos, which are like little genes that help knock down a target gene of interest, maybe related to a magnetic sense, like ferritin or so. Can we knock that down and affect their behavior? And the genetics here, just we can kind of at the same time track what's happening genetically. But ultimately, we want to see if some of these outcomes will affect behavior, right? If we get rid of magnetotactic bacteria, do they lose their ability to orient? Question.
What happens if they overproduce it though? Will they become more sensitive to the perceived electric or the magnetic sphere?
You're saying if they have more magnetotactic
Yeah.
bacteria? I mean, we really have no idea, right? We don't know yet how we could help encourage them to make more. But what we do know is we can supposedly disrupt it, right? With antibiotics, we can get rid of the bacteria, but yeah, we don't really know how to inject them with more without them, their immune system's killing it or whatever. So yeah, I'm trying to, again, we're starting from somewhere. This is very early in these type of studies, and we'll see what happens if we can get funded to do them. But yeah, so ultimately that's it. So I wanted to share with you today. Just want to thank my lab group over here, as well as all the different collaborators that have helped with all the various different projects that I presented on. And part of the reason I'm here is I'm right down the road. I do all sorts of genomics work in various ways, and I'm looking to kind of expand and do more work that might relate to space biology. And talking with some of the people here, we've already started a few little projects or talking about the projects, and always looking for new collaborators. So if you ever want to chat about things related to magnetoreception and how that might relate to magnetic environments outside this planet, happy to chat. So thank you very much.
Question.
Yeah.
So in one of the pictures, it went from, I think it was an iron radical, like a positive just iron, ion. Could you essentially make like a bioreactor, use like a big tub of water, essentially, and a fancy version of it? And our simulant is very iron rich. Throw in a bunch of that and they would just produce the iron in themselves, oxidize it themselves, and essentially separate the iron from the simulant, and then you can just collect the iron from them. Over.
Maybe you're getting pretty advanced for what I'm thinking. You're talking about where we have this protein ferritin that actually oxidizes iron to store it, right? Or you're talking about bacteria taking in iron and turning it into well, they're also oxidizing it and making those chains. Is that more what you're referring to?
Like using the bacteria to essentially separate the iron out of a mix of different things.
At a very first approximation, yes, right? They do take in iron from the environment, oxidize it, and store it as magnetite. So theoretically, they would be able to separate it or at least pull out some.
Because our current efforts right now are like heating it to 1000 C and then trying to separate it that way. That uses a lot more energy than that work.
Yeah. Well, if we could get magnetotactic bacteria to enjoy the various lunar simulants or lunar soils, you never know what's possible.
Yes. The lobsters, don't they have little like antenna receptors?
Mhm.
Why didn't you look at that? Or is it?
Yeah, I mean, there's a lot of potential tissues we could look at, and we started with their different ganglia because that's their largest unit, their largest clusters of neurons. So we thought that was a great place to start, and they're more analogous to the brain of the fish, for example. But yeah, I mean, the other sensory apparati, right, like the antennae, which is more of like a chemoreceptor, at least as far as we know. I'm not an expert in all the sensory physiology of spiny lobsters. Might still be really interesting. Yeah, we just don't know, and more experiments need to be done.
So MTV, are they everywhere?
Yes, which is also the hard part, right?
Yeah.
It can be kind of in some way contaminating everything.
Yeah. And so that brought me to the question about the tiers, and then also that the NCBI data,
Mhm.
right? You're getting samples from, from all these labs and different tissue types and different procedures and stuff. So how worried are you that a lot of what you're getting is just contamination?
Yeah, so at least with the current sea turtle swabbing is we also collect immediate water samples, right? So we
Okay.
can at least describe
Yeah.
the MTV community in the water.
Yeah.
And then the turtle, and we kind of have our background level then, right? So what the unique parts of the turtle might be part of the turtle itself,
Mhm.
or associated with the turtle. For the database mining, it could be contamination, right? We find these at very low frequency. What we're trying to, when I mentioned the second phase is trying to figure out what the patterns are. We're
Mhm.
trying to see if there are these patterns of species-specific bacteria, right?
Mhm.
If it was contamination from the environment, we would probably see more general things occur in almost every sample or very common and kind of more ubiquitous,
Mhm.
or depending on the environment around those samples. But if we see specific lineages or species of MTB that are only in, let's say, reptiles, but not found in other ones, and we see that occur many, many times, I think we start to remove the effect
Yeah.
of just contamination.
And then what about tissue type? Are you concerned at all or thinking about tissue type?
Yeah, right now we're trying to describe just anything that's there, but yeah, later on it's like, does it vary in tissue, right? Is it
Right.
in the eyes or in the gut or
Yeah.
so, but.
Because that might also help pull out some of that contamination.
Exactly, yeah. Right now, most of the microbiome data are from guts, right? And so the
Mhm.
contamination is possible. It's also could just be from their diet, right? They're eating something that had soil particles, that had magnetotactic bacteria, or in the water they're drinking, which is why we're trying to now get around gut samples, right? So
Mhm.
other types of tissue where we can kind of better control whether or not it's been exposed to environmental MTB contamination.
Mhm.
Yeah.
So where do you usually get your funding from?
I trial out with NSF. This is a lot of the basic biology questions that I work on. It's obviously very, very competitive, and I'm trying to branch out and explore how things I do can relate to these other funding opportunities. So
Do you have an Adco's Office of Scientific Research there?
yeah, so a lot of the early magnetoreception stuff, and this was actually done when I was a postdoc at Duke. We were funded by AFOSR for a lot of the mechanistic side of magnetoreception. But yeah, so moving into some areas related to NASA and some of their interests, talking with Stephen, right, we're looking at space. Well, I already submitted a proposal to space biology, but not magnetoreception. It's some whole other exolith related questions, but maybe some exobiology programs for some of the other exolith questions we have. I'm sorry? Yeah, so we submitted a proposal to the last space biology call. They had a call for animal studies, basically looking at the effects of simulated lunar regolith on biological systems. And so we proposed using a study on pill bugs. Is anyone familiar with pill bugs? There's so many different common names: roly-polies, pill bugs, potato beetles. They're small crustaceans, actually. And sorry. Yeah, you're right. So there's a variety of different species. They're a small crustacean, a terrestrial crustacean, and they're actually really great pioneers of new environments. So they've spread, at least one particular species has spread throughout the entire world, just because humans have introduced it everywhere. But they're really great at digesting all sorts of plant material. They're really hardy. And they can live in a variety of soil types. And so, and again, since they pioneer all these new ecosystems, we thought they'd be a good model to use to expose to lunar regolith. As essentially, if you're going to have closed ecological systems on the moon, for example, you're going to want biodiversity that can do a variety of different things. And so these pill bugs can digest a lot of material, human waste, plant material, etc. And they also can deposit heavy metals into their shells to kind of store it or remove it from the environment. And if they can live well in these lunar soils, then maybe it's a good biodiversity item to bring with us to these new areas that we colonize. So that was the proposal we wrote, and see what happens. Yeah, so that's the space biology.
I guess on the same space topic, I'm looking at the magnetotactic bacteria in space. And it has flown and they've studied it. But have you done experiments where either you put it in a microgravity or apply it to the ISS?
Yes. So I wouldn't call myself a microbiologist, despite working on these magnetotactic bacteria now. I focus more on the genetic evidence of them and where they're found and these type of questions. I don't manipulate them in the lab. Probably, yeah, just not the right person for that. But to work with people who do that, maybe there's some cool new questions that we could ask related to that. But yeah, I know there are people who actually study these bacteria from the kind of industrial perspective that have been thinking of, space flight and these kind of things. But yeah, I think more about how they are involved in host organisms and the biology.
Really exciting. I'm glad I got to learn finally about all the different things you do, maybe not everything, but I love your spiny lobster idea because it involves behavior and the live lobster afterwards. So how are you going to assay the sort of magnetotactic behavior of the lobster after if it's doing like a circannual migration or a lifetime migration to adult grounds?
Yeah, so so in the lab, there's a couple of different procedures that we do, and I've worked with colleagues who've done the spiny lobster work, the behavioral side of things, and I've done it in fish and other things. So there are two assays. One is a preference assay. So this is all controlled, aquatic, container basically. They have two different burrows on either side of their cage, essentially concrete block with a hole in it, and the lobsters love it, right? They feel very protected or so. And you can put a really strong magnet on one end and a mock magnet on the other end, at the same mass or so. So basically one burrow has a really, really strong magnetic field associated with it and the other one doesn't. And there's a really large preference for the one without the magnet.
Okay.
And you can basically manipulate that as a behavior, right?
Hey, nice.
And so that's the preference assay. The other one are directional assays. So you put them in a circular arena and you essentially release them in the middle. You can put little goggles on them so they're blind. So you release them in the center of the arena and you record the direction they travel. And I actually developed R packages for statistical tests of circular data. And you try to look to see if their direction preferences change under different exposures, right? So before and then after magnetic pulse, or maybe before and after treatment with antibiotics, you can see if their directional preferences are altered. So those are the two kind of behavioral assays we do in the
Can't wait to
lab.
see this.
We'll tell NSF to fund it.
Call me down because those lobsters.
Yeah, so Keys Marine Lab has all that set up. We just go down there. Oh, sorry, I didn't say it. No, that's fine.
Be a bit out there, but if there's any bacteria inside of humans, I mean, is there like, I don't know, are there like humans that are maybe more sensitive to magnetic fields than others?
That's debatable. That's a,
Is that something that-
yeah, so there's one group, a really kind of old school but well-known person who studies magnetoreception, a physicist at Cal State something, I forget which one. But does a lot of work on potential for human magnetoreception. And they're able to put humans inside essentially Faraday cages to prevent any sort of RF interference or so, and look at and record brain waves. And then they can manipulate the magnetic field in 3 dimensions around them, which we do on a small scale, which what Annalyn we're talking about building one ourselves. You can manipulate the magnetic field around them, and there will be slight changes in the brain waves. And those are claims of human magnetic sensation. But then there's a lot of debate on whether we're actually perceiving it, even though our brainwaves might be altered, are we actually perceiving and using that information for anything? And does that count as a sense or actual perception? So we don't really know, but human brainwaves are altered by magnetic field around you. Now, we don't know yet about how the magnetotactic bacteria that may reside in people are affected yet. We haven't gotten to that point, but yeah, the human area is quite debatable on whether or not we have a magnetic sense. Here first.
Have you thought about how MTB becomes more concentrated from juveniles to adults? So, I mean, obviously you can't look at it with sea turtles because you probably can't sample a juvenile, but they probably have the ability to some sort of magnetic field or follow some sort of magnetic
Yeah,
field.
so maybe, are you thinking more like vertical transmission of the bacteria? Is that kind of what you're getting at?
Just how it accumulates in its biome. It's
Yeah, so Juliana, so one of my grad students, I showed you a picture. She's actually interested in, well, doing all this work with sea turtle magneto microbiome. She's gonna have samples from adults, juveniles, and we're thinking of trying to get permits for sampling the eggs after they hatch. We're trying to see if we find the same magnetotactic bacteria in all 3 stages.
That's exactly
to
right.
suggest potential vertical transmission versus are the sea turtles just picking it up from the environment, right? Which might happen as well, but we're trying to kind of separate out those 2 possibilities. I think, sorry, back here.
What is the time scale involved in forming the magnetic receptivity or deforming the magnetic receptivity?
In which species or which organism or?
What is the time scale involved in these?
So we don't fully understand the question. For a bacteria to make a magnetite particle from iron, I actually don't know. I don't know how long it takes. Yeah. So,
But for example, I'm interested in solar storm part actually, because people use solar storm to predict the misguided birds migration and things like that. So they are short-lived. Actually, solar storms affect the Earth magnetic field maybe few hours. Actually. The reason I'm asking this question.
Yeah, so I guess I don't have the right answer for your question, but we actually published papers on how the solar storms are affecting our orientation experiments in fish. So when we're recording, we might pulse magnetize the fish, see how their behavioral directions change. And occasionally we get really weird results. And those results highly correlate with solar storms that happen at similar times.
Yeah, some of the solar storms, they have misguided whale spawn actually, and then migrating reptiles.
Yeah, other work I mentioned too with solar storms affecting whale navigation.
Yes.
Because we think some of the RF that might be more likely to occur at that time is interfering with the magnetic sensor because they interact.
I have another question. When earthquake takes place, the magnetic field in some certain region get affected. Is that has been recorded in this way?
You mean like the secular variation in Earth's magnetic field? Yeah, so we tend to think that there's a buffer in the magnetic sense of many animals. It's actually not incredibly precise. And so that helps buffer against the secular kind of day-to-day or short-term changes in the magnetic field. But when you get these longer term drifts, like the geomagnetic north that shifts over the last 100 years, several hundred kilometers, right? So like those kind of long-term shifts will overall have an effect. but on the day-to-day basis, the sense is not precise enough to return you to an exact location, but it might get you to a large enough area that buffers around what the variation in Earth's magnetic field will be.
Earthquakes,
Sorry, what's that?
yes, earthquakes.
Oh, sorry, I didn't hear about earthquakes.
Earthquake actually causes some disturbance in the magnetic field.
Mhm.
Because of the some iron core moves and that causes magnetic field disturbances.
Yeah,
So
so
was that has been recorded or?
yeah, I'm sure it has. I'm not aware of the literature in that area, but they would be very short-term effects, right? And so maybe the birds navigating through that area or other organisms moving through that area that use a magnetic sense might be disrupted. I haven't seen any literature on it, but I haven't looked.
Okay, because this is something related to geophysics. Actually.
Mhm. Oh, yeah, no, there's a lot of
For
relations to
example,
geophysics, yeah.
we talk about anomaly in the earth magnetic field, and the anomaly also moves. Actually. For example, the magnetic equator constantly changing. Actually. The earth magnetic equator.
Exactly, yeah, and a lot of the work in whales is that we think that they're actually following some of the anomalies that are occurring as part of their long distance navigations. Yeah, so the magnetic field seems to be really good for navigating at very long distances, but then when you get to roughly where you want to go and you need something more precise, the geomagnetic field is not great for that. But it's really stable over the course of the lifetime of any one organism.
Thank you.
I think did you still have a question?
Sure. With regard to humans, is it possible that at some point in human history, humans have worked completely into magnetic field when modern day they just don't have that. So we don't see it in any kind of.
Sure. I don't know the answer to that. One thing that comes up is there's some interesting evidence recently that during the last semi-magnetic field reversal, though it's called the Lechamp event, it was a near reversal of Earth's magnetic field. So it got really weak. There's probably some effects on the biological systems. It correlates with the time when our ancestral humans, we believe, were spending more time in caves and we're actually protected from some of the effects of that polarity reversal. It's, a little bit of evidence, a little bit of kind of hand waving arguments, but there might be some relationships with human history to that event. It's probably the closest I can get. But whether or not we had a better magnetic sense or orientation capabilities many generations ago, I really don't know.
The bacteria, when they separate the iron and reform it into magnetite, right? And then do they expel that, or do they just continue to grow?
So there is a limit. I don't know what it is, but at some point that magnetosome, that organelle that stores the chain of magnetic particles, is probably going to be too large for them to perform the rest of their normal functions. But you saw, like in one of those pictures, there's some very long, thin cells, bacterial cells with long, thin chains of magnets in there. But they can also expel it and not have any chains of magnetic particles in themselves as well. So they don't have to have it.
Some great discussion, everybody. Let's give another round of applause for our fantastic people. Thank you for having me.